In this video, we're going to discuss DNA structure as part of the second key area now, DNA structure and replication, all part of the human cells unit of the higher human biology. So DNA is made up of its basic building blocks, nucleotides. So it's thousands of these nucleotides all joined together, which are made up themselves of three key components that will come together to make a double strand. Bases will pair up in certain ways in the centre with a sugar phosphate backbone on the outside. And because DNA is double stranded, we also will have to take into account directionality, so the anti-parallel nature where the two strands run alongside of each other but in opposite directions. So we'll break down the structure of DNA into its key components. So like we said, the basic building block of DNA is the nucleotide, which has three key parts: a base, of which there are four varieties, deoxyribose sugar, and a phosphate. So it's good when we're looking at the nucleotide to consider the carbons as well. This will help out later on when it comes to how these nucleotides join together. So for numbering the carbons, we always start carbon 1 at the base and then go in a clockwise direction from there. So it's carbon 2, carbon 3, carbon 4, and carbon 5 being outside of the ring structure where the phosphate attaches. Remember, DNA is double stranded. It will ultimately form a double helix, a spiral shaped molecule. So, of those nucleotide components, sugar and phosphate are going to be on the outside, forming the backbone, with the bases always pointing toward each other in the centre. So as these bases line up in the centre of the molecule with the sugar phosphate backbone on the outside, remember they will always align in very specific ways. So A will always be opposite a T, adenine and thymine, whereas C will always be opposite to the G, the cytosine and the guanine. And what keeps them in that position, what keeps them bonded together, are hydrogen bonds. We mentioned at the start as well the anti-parallel nature of this double-stranded structure, which is all to do with the directionality and where the nucleotides attach to each other. So if we start with one strand, the 5' end is where you have your phosphate. See how there's nothing above. This phosphate is not attached to any other nucleotide, whereas at the opposite end, you'll have the 3 nucleotide at the 3' end. Okay? So 5 and 3 must always be opposite to each other. So on the opposite strand, this will be your 3' end, and because there's nothing on the carbon 3, it's not got another nucleotide above. So that leaves our opposite end is 5'. That's how 5 and 3, whether up and down or side to side, are always opposite. So again, we've got our phosphate group here with no nucleotide associated. So let's try our past paper questions, thinking specifically about the structure. So in this question, we're told a fragment of DNA contained 144 nucleotide base pairs. So what is the total number of deoxyribose sugars in this fragment? Pause the video and choose your answer. The correct answer here is D. So remember, it's 144 pairs, so 144 nucleotides in one strand, 144 on the other, and each nucleotide will have its own deoxyribose sugar, So it's a total of 288. Let's try another. Okay, the diagram will show us a part of the process of DNA replication, which will be a topic of a subsequent video. But it's good to see how knowing the structure and appreciating the different features of the DNA molecule can be useful for what's to come later. So here you're asked to name the type of bond which is linking the primer made of those nucleotides to the strand B, and also to name the chemical group that's found at the 5' end of any DNA strand. So again, pause the video and write down your answers. Okay. So for question A, which is the bond that holds the primer to the strand, remember it's the bond that is linking the bases together, just the bases are appearing in the primer, which is a whole series of those nucleotides. And you have to be specific, it's hydrogen. H just represents a hydrogen atom, but you have to specify it's a hydrogen bond. Okay? Anytime you've got two bases coming together, they'll be linked by those hydrogen bonds. And the chemical group found at the 5' end, that is your phosphate. Again, try to write it out in full here. Just writing P or PIs we saw in the respiration topic, the inorganic phosphate, is representing the atom and not the chemical bond. So between bases are hydrogen bonds, and phosphates are found at the 5' end. So good ideas to practice drawing out these nucleotides, try building up two strands of DNA. Knowing the structure will help out with the replication. Any questions you have, feel free to leave.